Okay, so team, when we were talking about cell cycle, I said I was going to kind of gloss over the cancer stuff, but then I was thinking that's doing you guys a disservice because it's freaking cool. You're not going to understand all of it, but that's not a problem because whatever, it's cool. I think you guys need to know about it. So let me go, I'll pull up my PowerPoint on my iPad. It's only like 2 or 3 slides. But it's cool and really interesting. So hold the phone. Oops. Okay, so it is at the end of chapter 12. All right, so here, and let me actually pause it just a minute, sorry. Damn, my phone is dinging, all sorts of things are happening. Hold the phone, sorry. Okay, all right here. So, okay, so we're right here on the cancer cells and how they break all of the damn rules, right? So, okay, so, yep. All right, so again, normal cells will, if there's a problem with their DNA, like a DNA damage or some issues, the cell will try to fix it. And if not, they will go through cell suicide called apoptosis. Cancer cells, on the other hand, if they have DNA damage shown by those little red X's, the cell will continue to divide, right? And that can give rise to tumors, if they are lacking that density-dependent inhibition. And if they have weakened adhesion, that can lead to metastasis. All right, and we talked about these cell cycle controls. Either we have these internal cell cycle checkpoints at G1, G2, and metaphase. And we also have these regulatory proteins, right? These cyclins, which cycle in the cell cycle. And we have CDK levels, which are constant throughout the cell cycle. And then we have this activity shown in green, that when cyclin levels go up, it will trigger mitosis, right? All right, and then we go on to this cancer, all right? So this is in chapter 18.5. There's only 2 real things that, that we have to know about, all right? And those are called oncogenes and tumor suppressor genes. And these are really important when we're talking about cancer. So oncogenes and tumor suppressor genes. All right, let's talk about oncogenes first. So oncogenes are overactive positive cell cycle regulators, right? So in the oncogene, if you have it activated, it is like pushing the pedal on the cell cycle, right? So oncogenes act as cell cycle activators, right? It's kind of like putting the gas on a pedal. So even if you're not ready to go through the cell cycle, this, if you have a mutation, will cause that to happen, all right? Tumor suppressor genes are the opposite. They are inactive negative cell cycle regulators. So what that means is that these proteins normally stop the cell cycle, but there's been a mutation that prevents that. So normally they put a brake. If they're mutated, they keep going, right? So tumor suppressor genes normally slow the cell cycle, but now speeding it up. So both of these proteins, if you have mutations in an oncogene, they will speed up the cell cycle. If you have a mutation in this tumor suppressor gene, they will also speed up the cell cycle. So one example of an oncogene is RAS. And RAS is one of the most important cancer genes in the human body. 20 to 25% of all human tumors have mutations in this particular oncogene. They're called RAS, right? And this RAS gene is a small membrane-bound signaling protein, All right, so it's shown right here in this kind of turquoisey color. It's membrane bound, and you don't have to know the details of the signaling process, but what you have to know is that normal RAS is involved in causing cell cycle division and proliferation, right? When there is some external source saying like, okay, now the cell's ready to divide and proliferate. In an oncogenic RAS, so in a cancer RAS, what happens is that RAS is always on, always causing cell cycle to continue, causing unregulated cell division, even if we don't have that external signal. All right. p53 is a tumor suppressor gene, all right? And p53, and it says, the statistics say that 50% of human cancers carry some sort of a loss of function mutation in p53, right? p53, again, is a tumor suppressor. So that means it normally stop the cell cycle if something's wrong. And if you have a mutation in that, then it's just going to continue, right? So p53 is something that you guys know about. It is a G1 checkpoint, right? It is a checkpoint protein that normally is involved in, again, using that stop if something's wrong, right? If the DNA is mutated, it will stop. However, in mutations in RAS, you lose that checkpoint and it continues to go even with damaged DNA. So here, if we look down here at the non-functional p53, what happens is that you get, even if you have unfixed or damaged DNA, you still get cell cycle progression. Whereas normal p53, if there's damage, the cell doesn't want to pass that on, right? And it'll stop and you can go through, the cell may go through apoptosis. So those are the only two I wanted to tell you about. I was gonna not have you know them, but I actually think they're really, really important. And there's something that students really think are quite interesting. So just as a reminder, I'm not gonna get crazy on testing you on these, but again, we have oncogenes, which drive the cell cycle, right? When they're mutated. And we have tumor suppressor genes, which often act as checkpoint proteins, which normally would stop. But again, if they're mutated, they're again going to drive the cell cycle. So those are two very, very common ways that humans get cancer, by mutations in either oncogenes or tumor suppressor genes. And both of those will drive the cell cycle, even if there's damaged DNA or other things wrong with the cell. All right, that's it. Stop that and bye.